Okay, guys, welcome back to the part 3 that is now. In the last part, we did all these means facilitated diffusion and all those, and today we'll be starting with active transport. Okay, we'll be starting with active transport. Now, some salient features about active transport is that it occurs against the concentration gradient. It occurs against the concentration gradient and it also occurs with the help of special carrier proteins called as pumps. Okay, these carrier proteins are called what? Pumps. I'm sorry for my handwriting there. These are called pumps. Okay, and they are also inhibited by the protein side chain. But wait, let me just make a chart. Okay, which will make them all clear for you guys. So properties. Now simple diffusion. This FD stands for facilitated diffusion and active transport. Okay, so this is a simple chart that I'm going to make. I'm sorry for my lines, guys. Okay, please bear with me. And this is the of I am drawing all vertical. I'm sorry. Okay, so the first property that we are going to compare is requirement of special protein. If special proteins are needed. Okay, so simple diffusion no, in facilitated diffusion yes, and in active transport yes. Let me draw a line. Why is not there? So I know maybe there is, I just don't know, I'm sorry. Okay, so let me just zoom out a bit and let me see if I can do this a bit straight. Okay, off. I think I'm going more bad. I'm sorry, I won't try this, but let's just make another column. I think this is legible, right? So I'll just make some more columns and yeah. Okay, so what is the second criteria that we will go with is specific. Is it, sorry, let me just rub it out. Is it specific in nature? This is a question. Okay, then the answer will be no, the answer will be yes, the answer will be yes, it is specific in nature. Okay, then can it be in can it be inactivated by protein inhibitors? Then the answer is no for here. It is yes, and it is also yes. Now, number 4, is it an uphill transport? Well, simple diffusion is not an uphill transport. FD, that is facilitated diffusion. Just remember the word diffusion is still there, guys. The D is still here. So it is also not an uphill transport. So no. On the contrary, we can write that this guy needs ATP. So we can write another one that if ATP is needed, and the simple answer is only active transport needs ATP. And the fifth and the final one is saturation of transport. Will it get saturated if there are way more molecules to be transferred rather than the, wait, let me just write, transport saturates, question mark, then no, yes, and yes. Let me just see the basic thing, the basic idea behind this is that since facilitated diffusion and active transport both needs help of special proteins. So what happens, say this is a protein, okay, and this protein, it's a protein, and this protein is needed for the transport of a substance, say A, okay, for transport of substances A, and for every, okay, let me just write P, for every protein P present, it can transfer only one A at a time, okay, it can only transfer one A at a time. Say this is the plasma membrane, just get my white pen, say this is my plasma membrane, and this transmembrane protein can transfer only one A at a time, so that it will bring that A into here. Now we have only one protein P, right? We have only one protein P, but three of A's. So what will happen? Or will all the three pass at the same time? Well, the answer is very well no, because since I've specifically stated that the protein P can only transport one A at a time. So what is this? Isn't this transport saturation, that this will go, then this, then this? Is the transport saturated? Say there are, I have shown only three, but say there are 10. What will happen? Won't be like, there won't be a traffic jam kind of situation. So can we say that the transport is saturated? Well, yes, we can, and that is exactly why in both facilitated diffusion and active transport, the transport saturation occurs, because they both need special proteins to function, and therefore their transport is saturated when there are way too many substances that are waiting to be transferred. Okay, so now let's move on, and this, I don't think, types of membrane, I don't think is much needed, but if you want to know, I can tell types of membrane, right? There are how many types of membrane? Now I won't go into very much details, but to be honest, I'll write there are four types of membrane, right? Impermeable, okay, impermeable membrane do not let anything pass through it. What are its example? You can write let's say, let's write an example that is present in our syllabus: cork and suberin. This is present in plants, right? Cork and suberin is an example of impermeable membrane, right? Casperin strip, yes, obviously. So we can say, then it is freely permeable. Freely permeable. Anything that comes to it will be allowed to pass. Can you tell me an example of this? Can you tell me an example? Okay, if you can, we'll just pause the video. I'm going to write it anyway. Cell wall. We will cover everything when we come to that topic called cell. Now here is the main problem that arises. Now there are two types, okay? Selectively or differentially. Sorry, let me just write this again. Selectively or differentially permeable, and there is another one that is called semi permeable. If there is too much confusion, you guys, semi permeable membrane. If there is, this is the one that is known as SPM, okay? If there is too much confusion, please just leave it, okay? Selectively permeable and semi permeable, okay? Now let's just write two examples. Semi permeable, Example is egg membrane. This is also written in NCERT, this example, egg membrane. Okay, and let's say selectively permeable membrane. Example is plasma membrane. I think you have heard of this. Plasma membrane. Tonoplast. Can you tell me what is tonoplast? Tonoplast is the single membrane covering of what? Sap vacuole. Okay, it is the single membrane covering of sap vacuole. It is related to sap vacuole, so I'm writing it here. Okay, and I think it might just be necessary. And okay, let me write out even this. Inner membrane of chloroplast and mitochondria. Okay, it is this simple. Please just remember that this, if it is very much, if you are stuck with it, you can just leave it or just remember, okay, this is all the things you need to know. And selectively permeable means only selected molecules can pass through it, nothing more. And the fact that the selectively permeable is also called differentially permeable, you just need to know that. And what is semi-permeable? In semi-permeable, solvent can pass but not solute. Okay, that is the only difference. Here only selected solute particles can pass, and here solvent can pass but not solute. Fine, fine. Let's go with osmosis. Okay, should I write? Yes, just let me write. Okay. In selectively permeable membrane, few solutes, few selected solutes can pass, and here no solute can pass, only solvent can pass. Okay, the difference is very simple. Only solvent can pass, no solute. So in this way we can say that selectively or differentially permeable, just this is an extra point guys, selectively or differentially permeable membrane is a special type of semi-permeable membrane. Yes, it is. Well, I have written it. If you want, you can just copy it. I do not know if it will be asked. Special. Okay, that is it. What there is to it. So if you want to know, you can very well know and write it. Okay. Now let's come to osmosis. What is it? Osmosis. Okay, let's come to osmosis. What is osmosis? Let's define it. It's a special type of diffusion of solvent across a semi-permeable membrane. Okay, across a semi-permeable membrane. Oh, what is this? Across a semi-permeable membrane. Okay, this is all there is to it. And if we take for solvent molecules, okay, remember what I said about water? Well, let's come to that part. In living systems, solvent is water. Solvent equals water. Okay, just remember this. In living system means the one we are concerned with. Now, osmosis can also be defined with the help of water potential concept, and I think you guys already know. I will just wait a minute, copy the diagram that I had done so long back and just paste it. Okay. Oh, what am I doing? I think I could have just drawn. What is this? No. So after all this, I just, how do you, oh my god, let me just delete it. Yes, thank you, and I'll just paste it. Okay, let me just do it a bit bigger, and let's say this is A and this is B. Let's do that same thing. These are solute particles and these is solute particles. So if you have seen that video, I think you will be able to understand what this is. This is a container filled with water. This is fully water filled, and these green particles, these green guys, these are solute.